Hello, everyone. So, can you hear me? It's good? Okay. Hello, everyone. I think that after the lunchtime is a good time to start to talk about fruit. Right, Mitchell? Like blueberries, strawberries. First of all, Dave, thank you for the invitation. It's a great pleasure to be here. Um, in my talk, I will not talk about a single project, but what I'm gonna try to make here is to show you how we are combining or how research and breeding they can run like side by side with different examples to support like breeding decisions. Okay. My name is Felipe. I'm research scientist in the blueberry breeding program at the University of Florida. For people that don't know me, do we have a mouse? No. Okay, here. Okay. So I joined the blueberry breeding program back to 2017, first as a postdoc, and I was hired. Patricio, that is the main breeder. He was just hired in 2017, and my main task it was like to help the breeding program to implement genomic prediction. And 4 years ago, I was hired as faculty in the horse science department. What is interesting about the blueberry breeding program at the University of Florida is that we have the main PI, that is Patricia, as I mentioned, but the organization of the lab is divided in four main areas. So we have here phenomics, statistical genetics, computational biology, and molecular breeding. And this is the way that the information flow across us. So basically, we have here data collection. I'm in charge of my group for make sense of the data. And then at the bottom here, we have biological discovery and gene editing and validation. So as you can see, everything that we are doing is to support breeding. This is the ultimate goal. In the specific area that is statistical genetics that I'm gonna mention, that I'm gonna present today, I'm working with two main crops, blueberries, and 2 years ago we started a research line with coffee. And what we can see is that we have different projects in the blueberry breeding program that we are using statistical genetics. So some of the crops it involves a kind of better understanding about some traits. So just to give one example, we have here, for example, this is a PhD thesis or from Julie, and she's trying to understand more about the genetics of Phenomocracy fruits. And we also have other kind of more theoretical studies, but again, all these topics at some point, at some level, we are using statistical genetics. And what I'm gonna try to present here is just some examples that we have in the blueberry breeding program. Okay, so let me start with the motivation. Why genomic selection? Why we are using molecular breeding in the blueberry breeding program? So the first one is what is blueberry? Why blueberry is so important? So blueberry is the second most important soft fruit, and in the past 10 years, we can see this increment or this increase in the consumptions. And there are two main reasons for that. Number one is because delicious. You should eat blueberries every single day, and also given the health benefits. On the market, the University of Florida plays an important role, especially on cultivar development. So nowadays, we release more than 60 cultivars from back to the 70s to 2023. And there are other 45 releases that are in collaboration or directly from our crosses. And there is a nice and clear philosophy in our group. There is breeding and research then can run side by side. So, and this is what I'm gonna try to present in the next couple minutes. This is the timeline. So, just a summary. So, how and when do we start to implement genomic selection in the blueberry breeding program? I like to divide in four main moments. So, the moment that I call it pre-genomics is when we, especially Patricia and I, back to 2017, we dive on those old field books that we had in the breeding program, old files, and we organized the entire pedigree, compiled all the phenotypic data that was collected, and it was the first generation of our pedigree, or the first step to start to use mixed models and to start to predict breeding values. So nowadays we have a pedigree that back like 70 years ago. There's more than 17,000 lines, and it's a very deep pedigree. So the next step is or the next moment is what I call it genomic selection 0.0. So it is when we decided to come to the company, design genotyping platforms, and start to thinking about or prepare ourselves training models to use genomic selection. So it took like almost 2 or 3 years to be ready and start to use genomic selection at the pace and scope of the breeding program. So 2020 was the first generation that we have like 20,000 plants that we never phenotyped, we just genotyped, and we start to take decisions based on this. Nowadays, we are in the so-called genomic selection 2.0, that we are just refining or trying to polish the protocol that we created like 4 years ago. Something interesting about this timeline is because at this part of the plot, you can see like a couple of papers that we released. But the most important is all those papers or all those studies, they were translated in practical applications. So it's one way to bridge this gap between breeding and also the university or research and breeding. Okay, so for in the next slide, so what I'm gonna tell you is a couple of questions that we did like when we started the first moment that I call it genomic selection 1.0, and then I'm gonna show you a couple of other questions that we have in the second version or in the genomic selection 2.0. So okay, so back to 2017. Those are the four main questions that we had. So the first one is genomic selection better than phenotypic selection? What are the best predictive models? What is the importance of better genomic resource? And can we unify prediction and discovery in a single framework? So to answer the first question, I need to tell you a little bit more about our breeding program. There's a very similar table that what Jeff just presented some time ago. So we divide in 5 main stages, as you can see here. So we start with every year, we make around 106 crosses. We put like 20,000 seedlings or 20,000 plants in a high density nursery. Those individuals, we're gonna select 10% using visual selection. And at this point, we still have like single information at the single level. Then after like 4 years, we select more 10%, and we start to put in a farm condition with the experimental design. So we clone the plants, and we have like 15 plants per plot. They're gonna be evaluated for more like 4 years. And then we're gonna start the final stage of the breeding program. So the best plants here, they are cloned. They're gonna go to the regional yield trial. We have like more than 10 farms across the Florida state that we're gonna test the best materials, and then we have the cultivar release. Okay, in a very simple way, in a very this is a briefly background about the breeding process, and there are a couple of challenges when we are describing this breeding process. Number one is time consuming. Blueberry has a high level of inbreeding depression and is an autotetraploid. So genomic selection can help in 2 different ways. Number one is we don't need to wait anymore until the stage number 3 to select the best parents to be crossed. So now when the plants, they have like 2 years old, we collect DNA, we extract DNA, and then we estimate it, or we predict the genomic breeding values, and we design the crosses. So this is one way that we can use genomic selection and we can maximize the genetic gains. Another way that we are not doing so far, but we can, is we can skip stages. So we can move plants directly from the stage 2. It's too aggressive, but maybe in the future we can make some tests. We can move plants directly from the stage 2 to the stage 4. So far we are using genomic selection to accelerate the breeding cycle and selecting parents in early stages. So again, to try to solve this question, is genomic selection better And then phenotypic selection, we published those 2 papers. I just want to mention that when we are applying genomic selection in polyploids, is a little bit different than in diploid, and those are the 3 main differences that we're gonna see. So we're gonna have more genotypic classes. So we need to estimate the allele dosage. To this end, we use the R package to create the relationship matrix. We use the AJ matrix R package that is fast, is user friendly. We can use like pedigree, genomics, and hybrid matrix. And something that is interesting is that the alpha difference than diploid is because we have more complex gene actions. So here is just one example that we have different levels of dominance. So those are the 3 main differences that I just like to point out when we are comparing genomic selections in blueberries, in diploids, and in polyploids. Okay, so back to 2017, we had a large breeding population, 2,000 individuals. We have different fruit qualities, and we tested fruit quality traits, and we tested 3 different models. One is a pedigree animal model that we call it ABLUP, a GBLUP, but we just assume that all markers are diploid, and then a GBLUP, but accounting for the allele dosage. So here's just a couple of some of the results. So only 2 traits I'm plotting here. So, and we can see that we have a better prediction ability when we use GBLUP and accounting for allele dosage. So this was one of our first take-home message. So number 1, genomics better than pedigree for most of the traits that we are looking for, and also on the relevance on using polyploid methods. A second question is, okay, so we're just looking for a GBLUP, but what are the best predictive models? So this is a work that we did in collaboration with Jeff, and here with some colleagues in Spain. The main question is if we can, most of the prediction models, they are based on linear methods and on additive gene actions. So the question is, okay, so can we use more elaborate models, for example, the Bayesian alphabet, and also try like different gene actions, and eventually using deep learning methods to incorporate non-linearity. And here we test, we make this question, and we test this for blueberries and also for potato. And the key result, the main point regarding this specific plot is that the additive GBLUP is doing a pretty good job for most of the traits in both species. Okay, so in a second paper, we decided to test not only Bayesian and mixed models, but also deep learning. And then here we have like 1, 2, 3. We have 5 traits, and we have 4 different models. So in blue, we try deep learning, and again, we can confirm that is on the relevance or on the importance of additive GBLUPs. 3 months ago, there was this paper that was published. There was a new paper using transformer deep learning architecture. That is the same architecture that was used by ChatGPT. And it's really interesting because the author tested the methodology in different data sets, and they made some strong claims. So you can come here and you can say that as a proof of concept, we show that this methodology is the first method which is able to consistently and significantly outperform classical methods in several databases. Okay, so we decided to test this. So we compare all the data files, it took some days to run, and the results is here is a summary of the results. So here we have the traditional GBLUP. The GBLUP has a accounting for the allele dosage, and this is the deep learning method. And once again, at least in our data set, using blueberries and coffee, we still it's really hard to beat the GBLUP for our data set. Okay. But this just confirm about the relevance of using or how solid are those methodologies. Okay. So Another question is, okay, so what is the importance of better genomic resource? Why we made this question? The central idea behind of this was published in this specific paper, but if we back to 2019, there was no reference genome for blueberries. The only thing that was available, it was a pool drafted, and there was a couple of challenge when you start to use a pool drafted. Number one is to design or to refine our genotyping platforms. Number one, there was a very poor gene annotation for gene mining, and it was really complex to design markers for a marker selection. The good news is because we started a collaboration with the Michigan State, and then we had access to a high quality and haplotype phase reference genome back to 2019. And what I'm going to show you is how this new reference genome changed our point of view about some traits. So let me give you a very simple example. This is the GWAS using, so we run a GWAS using this, what I call it, full genome or the drafted genome. And then it's exactly the same GWAS, but then all these SNPs, they were called using the new reference genome. So we could find more association. But what was really interesting is because when we run, so this is one specific volatile, and when we run the GWAS analysis, we saw a lot of hits. And our first idea is, okay, it seems to me there is a very complex trait. But then when with a better genome, when we could organize everything, we could see that all those hits from different scaffolds, they were coming from only two main regions. And this change, so what I'm telling here is we had a much better understanding about the genetic architecture of different traits, only using like better genomic resource. So we also decided to refine or to reduce the number of probes only using bioinformatics. So with a new genome, we just took a look on the probes and we tried to reselect the number of probes. And then we run genomic selection, just looking what are the probes, what are the best probes, how the probes they are aligning with the different homologous. And what we can see here in black is the prediction ability using the new reference genome, where we redesign our probes. And in white, we have the old prediction ability using the draft genome. And we can reduce, so here is using 30,000 probes. And with the new genome in black, only 10,000 probes. So we reduce 3 times the number of probes and then reduce the cost as well. And we have pretty much the same or better prediction ability. So this is another take-home message. So good genomic resource can help us, can help contribute. So the last question in what I call a genomic selection 1.0 is, can we unify prediction and discover into a single framework? This was published in this paper in Phenomics a couple of years ago. And here we are talking about a complex trait that is flavor. And the central point about flavor is because what we recognize as flavor is the combination of two main things, the taste and the aroma. So every time when we eat any kind of food, so our tongue, receptors in our tongue, is going to recognize what is sweet, what is sour, what is salty, bitter, and umami. But then there is a group of other feelings that is going to come from the aroma. So the fruits, they're going to release what is called volatile organics components. And this is going to give us the perception about the aroma. So the association that I can make here is what happens when you are eating a food and you have a flu or you are cold. So your perception about the flavor is not the same. And the main reason is because you cannot realize about the aroma of the food. So humans can recognize more than 1 trillion of different aromas. So aroma is really important to define the flavor of any food. Okay, so given the importance, our breeding program is looking in more details about those VOCs or those volatiles, because we are confident that this is one way to understand this complex trait that is flavor. Okay, So here we have another point that is really interesting, is because we have a lot of evidence that most of those volatiles, most of those metabolites, they have a simple genetic architecture. So this is one volatile that is pretty common in blueberry. There is called 2-andecanone. And what we can see is that when we run a GWAS analysis, we have a significant SNP covering to a tower-like structure like this. So this marker here at the top by itself is explaining more than 30% of the variation on this volatile. So it's a kind of simple trait controlled by one or few genes. So then we decided to make genomic selection for volatiles for aroma, but trying to combine biological discovery in the same pipeline. So how? So first of all, we have two different populations here. One population that is in light gray that we just use for GWAS. Okay. Then we decided to go to the field and collect a new population that we call it as validation. So this validation was never used in the GWAS framework. This is a true validation. Okay. So, and then we designed like different scenarios. So the number one is when we come to this first population that I call it as the original population, we test and train our model inside of this population, and this is the prediction ability for different kind of aromas that we have in blueberries. Okay, so then we try something different. So we train the model in the population one, and we test and we validate it in the population two. And as we might expect, the prediction ability it was reduced because these are kind of true validation. In another scenario, we decided to do something different. So we run the GWAS in the population number one, and following some ideas from Rex Bernardo, we use the hits that we found in the GWAS analysis as a fixed effect to predict the population two. And what we can see is that for most of the traits, when we compare the yellow and the green bar, is we have an improvement. And the last scenario is the most drastic one. We just make predictions. It's not a genomic selection model anymore. It's just we call it Marcus's selection because we just got it the hits that we found in the population one in our training population, like few markers to predict the population two. And we can see that, yes, it seems that it's a very simple trait because only using like 2, 3, 4 hits or 4 markers, we have a very good prediction ability. Okay. So the take home message is if you have like prior biological information, this can be used to improve the prediction ability. If you have like traits that they are simple as volatile, using this approach, it could be a good idea. Okay. So then, with those 4 main questions, we have the present and the future in blueberries that involve different kind of studies. So we are testing like new methods. We are investigating new traits. We are implementing metabolomic selection at the pace and scope of the breeding program. And now artificial intelligence is everywhere. But what do we... The 4 main questions that we are trying to solve in this new generation of genomic selection that I call it genomic selection 2.0 is, number one, can we optimize our breeding program by reducing the number of markers and optimizing our training set? Is multi-omic predictions a good alternative in a practical breeding program? Number 3, what is the impact of Phenomics on food quality prediction? And the last one is how artificial intelligence can shape the future in a modern breeding program. So to try to answer those questions, let's start with the number one. Let me give you a background about how we are using how we are genotyping our breeding population. So the genotyping platform that we are using, the blueberry breeding program, relies on target capture sequencing. So we started back to 2017 with 30,000 of probes. And basically what is probe? Probe is a region that we're gonna target in the genome. So, and those regions, we can design those probes using different parameters or different ideas. So when we originally designed those probes, we try to maximize or we try to capture, number one, genomic regions, and number two, have access to the entire genome. So we try to design probes in this way. As I mentioned to you, with a new reference genome, we refined this, and now we are not using 30,000 more. We are using 10,000. But then we want to try to see if we can reduce still more. We can have like less probes, for sure, to reduce the price of the genotyping. And what is interesting is because each probe, they're going to output different results. So we have probes that we're going to have more SNP, and also some probes, they're going to have different quality parameters. Okay. So what we decide to do is, okay, so what happened instead of to randomly select probes and see if we can keep the prediction ability, what about if we use a kind of sequential methodology? So the first layer of information that we use to filter the best probe is, again, we know that some probes, they're going to have more or less SNPs. They're going to capture SNPs more or less a minimal allele frequency. So we decide to rank the SNPs based on allele frequency, call rate, and number the number of SNPs per probe. So then we run the UpDog, and the UpDog is a good tool because the UpDog is going to output some parameters. Then we can select probes based on the bias, based on over-dispersion, and sequence error that is output from the UpDog. In another layer, we compare the genomic relationship matrix that we got it per probe with our original pedigree. So if a given probe measures something that is very different than we have in the pedigree, we decided to remove. We have another layer that is called repeatability. We have some individuals that we have multiple data that was sequenced like 5, 6, 7 times. And it's very interesting that for some probes, every time that we sequence in the same individuals, for some probes, they are giving us like different genotypes. So we call this repeatability. And then we use an extra or an external breeding population. Actually, it's a biparental population. And we saw that consistently, some probes, they have a high segregation distortion. So then we decided to remove this. So we put all those information in a classical selection index to ranking the probe and to select the best probes. So here is the original number of probes, 10,000, and how they are distributed across the genome. Then using 5,000, using, of course, this selection criteria, and then using 2.5K probes. Okay, so what you can see is that, okay, so we have a drastic reduction, but are still covering the entire genome. And then we run genomic selection, traditional genomic selection. And basically, when you compare all probes with the reduced number, we have a very, very similar prediction ability. But again, now we are moving from 10,000 to 2.5K. So it's a big reduction in terms of cost. Another question is, what about the training population? So how can I optimize my training breeding population? So one of the key point is our first genomic selection model was trained back to 2000. I mean, the data was collected in 2014 and 2015. We have 2,000 individuals, but now we are trying to predict individuals that they are like in 2023. So using this massive number of individuals and phenotypes that we collected like almost 10 years ago, the question is my old model still calibrated? Should I keep using this information in my to train my models, or I can just exclude those old phenotypes and just include like new genotypes? And if the idea is every year, for sure, we are including new genotypes, but the question is, how many new individuals should I include in my training data set, and how I going to select those individuals? So, for example, I can go to the field and I can select, for example, one family, or I can phenotype 2 families, 3 families, or few individuals across multiple families. So what is the best way to select individuals to keep updating my genomic prediction model? So the first result that I want to share with you is, so here at this point, we have the prediction ability when I come to the population in 2022, I phenotype a genotype, I run in a cross-validation, and then this is the prediction ability in for those three different traits. Okay. So what I have here is what happened if I come to my historical data set, the data set that I collected in 2014 and 2015, and I train my model using these data and try to predict a completely new population that we have in 2022. And the answer is yes. So even using this old data set and that we collect like 10 years ago, they are still giving us like good prediction abilities or positive prediction abilities. So there is no reason on all the tests that we did to put those data in the trash, even if they are like 10 years old. Okay, so they are still part of our breeding program. But our question is, okay, so how many individuals I should include on this large data set that we have to keep improving the prediction ability? So what you can see here at this bottom, we tried different algorithms, different methods, and then we included in this historical data set like 100 individuals, then 200 individuals, then 300, 400, and 500. And what is interesting about that is there are a group of methodologies or approach that we can see here in the model. They are algorithms that they try to maximize the genetic diversity. And when we maximize the genetic diversity in our training data set, it's a good idea to be included in your training data to predict a new generation. Okay, so one of the take-home message at this level is we can reduce the number of markers, but also we can optimize the number of individuals that we're going to use in our training population to maximize the accuracy. Okay. A second question is if we should rely only on genomic prediction, or maybe if we try to combine multi-omic to predict like complex traits. Okay. The whole story about this was published in this paper a couple of years ago. And the central idea behind of this is that, again, evaluate flavor is not simple. So basically, the best way to evaluate flavor is by providing consumers with a sample set of the food, and then we ask their opinion. So basically, here we have a real consumer, here we have a blueberry sample. So the consumer is going to try the blueberry and it's going to tell us, okay, so what is your score for overall liking? What is your score for sweetness, sourness, and different traits? There is a big limitation with this. Number one is expensive, time consuming, and low throughput. So a single sensor panel in the University of Florida costs like 5,000, and we can include only 5 genotypes. Why? Because if I give you more than 5 genotypes and you start to eat blueberries, after 5, you're gonna say that everything is the same. Okay, so it's the most expensive phenotype that I know. So we are paying like 1,000 to phenotype a single genotype. Okay, so it's very expensive and low throughput and a lot of noise because most of the consumers sometimes they cannot understand the scale and they can mess up the process. Okay, so one alternative is what happens if at the same time that we are running the sensory panels, we send the samples to the lab, and in the lab we're gonna collect the sugar and the volatiles, and then can I predict how much a consumer is gonna enjoy a berry, but just using the sugar, acids, and the volatile information? So this is the question. If you give me one blueberry, and with this blueberry, if you can give me the volatile profile and the sugar and acid profile, can I predict how much you're going to enjoy the berry or what is the aroma of the berry? And this is the key point behind of this research. So to test it, we used 2 different models. One was tomato, another one blueberries. We used a historical data set, large sensory data that we have in the University of Florida. So we have more than 2,000 of consumers here, more than 3,000 of consumers. We have different volatiles. We have also sugar and acid. And the sensor idea again is try to predict the sensory or the consumer preference just using the metabolites. This is one of the key results. Here we have different statistical models. Why we test different statistical models? Because in the food science, the most common is the partial least square. So they love to use partial least square. So this is our contribution for the food science. You know, we have some other methods that eventually can do a better job. So we have here the two different species, blueberry and tomato. And here we have a different trait that the consumer, so we ask the consumers, for example, at this point, okay, give me a score about the overall liking. So what I'm telling you is that if you give me the volatiles and the sugar and acids, we can predict using random forest, for example, with more than 60% of accuracy, how much a consumer is going to enjoy a berry. So if this is true, and this is true, so we can replace, we can use this information. We can use those predictions to know how much a person is going to enjoy a fruit. Okay. So in the case of we try in the case of tomato, we have a whole genome sequence for a couple of genotypes. Then we compare the metabolomic selection against the genomic selection, and here's the prediction ability. So when we try to predict sensory traits using metabolite, we have a better prediction ability than using genomic information. Okay, so 2 years ago, I decided to run a similar trial, but in this case, we had like 1,000 of different blueberries, and myself and another breeder, we decided to weigh 1,000 of blueberries and score for aroma and liking. So don't ask me about the result. It was not. Yeah. So we evaluated this, and then we run the prediction ability, and it's really interesting. So this is when I try to predict the liking scores of those two breeders using, for example, only traditional fruit quality traits. When I say fruit quality, it's like only Brix and acidity. But when we start to expect like metabolomics and genomics, so you can see what happened with the prediction ability, just increased. So this is aroma. So if you want to put your fruit in another level that is not only sweet or acid, but if you want to bring like different aromas, like floral, sweet aroma. So if you rely only on fruit quality traits like Brix and acid, is not enough. So take a look what happened when we use the volatile to try to predict aroma. And then what happened when we combine genomics, metabolites, and also the traditional food quality traits. So we can have a very good prediction ability. So the take-home message is multi-omic is a valid tool for predicting complex traits. Okay. So we also test the idea of phenomics. This is the first table that I mentioned to you, and there is a huge bottleneck in the breeding program because at this stage, so those are the selection criteria that we use to move plants across the different stage, and that at that point we use visual selection. So basically, we have 20,000 plants in the field. We have 2 or 3 breeders and field technicians. We need to go there and try berry by berry. So it's really hard. So what about if we change this, you know? So using genomics at this level, I mean, genomic selection is too expensive, but what about phenomic selection? And the central idea is try to use NIR. So there is a non-destructive high throughput method, and it's based on the absorption of the near infrared region. And what we're gonna have at the end is this wavelength that we can treat as a marker. And based on this is one example in soybean. So just based in the same way that we have like, imagine that each one of these lines here is a wavelength. So the association that you can make is imagine that we have like different markers. So what happens if we created like relationship matrix based on the NIR and not only in the same way that we use genomics. So Here we have a nice result. So we have 5 different traits that we tested: NIR, pedigree plus NIR, genomic plus NIR, and genomics. And we can see that for a couple of traits, for example, bricks, when we try to predict only use NIR, the prediction ability is better than genomics. Okay. But what is really interesting is because when we start to combine information like NIR plus pedigree, genomic plus NIR, when we start to stack information, we can increase the prediction ability. But just to be careful, NIR, it seems a nice tool so far, but it's very sensitive to GBI. So if I collect NIR in 2022 and try to predict genotype in 2023, the prediction ability just go down. And also it's very sensitive to the tissue that you are using. So we test this in the whole berry, in the leaves, and also in the juice. And the prediction ability is very, very different. So these are work in progress. I just want to show you a kind of snapshot about about some of the results. But at least at this moment, again, this is a working process. It seems a good alternative to genomic selection on early stage. I just have 5 more minutes. I just want to mention how artificial intelligence are changing our breeding program. So far, we have focused a lot on collecting different data sets like phenomics, genomics, and metabolites, and also testing different classes of models. But the question is, can we collect better phenotypes using artificial intelligence? And this is one example. So here, this trait is called wax. And here is a blueberry with a high amount of wax and a low amount of wax. And traditionally, we are measuring this using visual scores, like ranging between 1, 2, 3, and 4. Okay. So then we decided to create a new pipeline or a new software that's called BRCV, and is based on computer vision, that we can train a model and we can quantify the wax. What is interesting is because when we use this traditional score, we never found a single hit in a GWAS analysis. Never. But then when we start to use the BRCV, there's the computer vision, you can see only in terms of genetic parameters. So this is the irritability. How is the difference in terms of irritability when you compare like the same genotype, but when we got the visual score and the computer vision? And then we could find 2 different hits that we never saw when we use visual scores. There are 2 different hits that we can move forward for marker selection in the breeding program. I'm gonna go directly to the 7 main lessons from the 7 years. My final message is based on all the results that we have so far, genomic selection works. We can have like solid gains when we decide to use data-driven methods. If you can, prefer to use like statistical methods that is designed for polyploid system. We like this quote from Leonardo da Vinci, that simplicity is the ultimate sophistication. So I think that make a lot of sense to test all those deep learning, machine learning methods. Make a lot of sense, but don't forget to include a GBLUP as a benchmarker. It's something that is really interesting. GS can be optimized after having a good understanding about the breeding pipeline. Knowledge is power. So all decisions based on multi-layers or multi-omics, it's important. And AI has an important play to role on data collection and analysis. This is my personal opinion about practical implementation in polyploids. So I'm just gonna read this in a very fast way. So genomic selection is a tool to assist the breeding and not the way around. So if you want to implement genomic selection, you need to have a very solid breeding program. The implementation, again, requires solid breeding program. Genotyping might be not so easy and cheap. So we have the PEG in, it's gonna start tomorrow. There are a lot of companies saying that it's cheap, but it's so don't believe that you're gonna have like good prediction models without good phenotypes. So sometimes we focus a lot on genotype, massive on genotype, but if you don't have good phenotype, if you don't go to the field and collect good data, it's not enough. Again, don't forget to use GBLUP as a benchmark. Is a multidisciplinary topic, and biological discovery and prediction can run side by side. Okay, so I have a lot of people to say thank you, especially Patricio, that is the PI of the blueberry breeding program, or the blueberry breeding lab. Those are a couple of talks from Blueberry that we're gonna join us in PAG. So a couple of talks and posters that we're gonna present in PAG. And thank you, Dave. Thank you, Jack, for the invitation. It was a great pleasure. 45 minutes.